Hello everyone, my name is Thu. Um, I will present about the animal testing. Actually, it is more about the alternatives to animal testing. But first, I want to show you this. This is my rabbit. She is very lovely and but she's stubborn and hot temper. But I never think that one day I can do something harmful to her. But I wonder that what should I do? What choice available if someday if my family member um, gets sick and let's imagine that there is a potential treatment available in the lab stage, but it needs testing on animal to ensure the safety before it can become commercial for the patient. What should I do in that case? So that come to the topic I want to talk about today. It is the whether any alternatives to animal testing. Actually, this topic already raised up and in late 19th century, but it becomes strongly controversial in the public as well as among the scientists in the last few decades, with the dividing into two groups, one supporting for the animal testing and one is again animal testing. Each side has their own reason to support for their arguments based on the pro and cons of the animal testing, and it covers from the ethical and the practical or effectiveness aspect, or practical aspect or effectiveness. So I prefer to go through the 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 development of the technology and the approach and methods that we have until today to bypass the animal testing while we still ensure the creditability of a research or a study or a test. So in general, the alternatives testing will base on the three R strategy. This principle is mentioned for the first time in 1956 by the two scientists, William Purcell and Rex Birch. It mentioned about the three different approach that is refinement, reduction, and finally replacement. You will find the more details about the refinement and reduction, what we could do in specific to achieve the reduction and the refinement as the alternatives for the animal testing in my slide. I would like to focus more on the third R, the replacement or the alternatives on the technology. The animal testing can be found in three different fields. It is in teaching, education, the dissection for the student, or in the testing and in the research. The alternatives in the education can include the audio visual materials and insensitive materials, or we use the models, or we use the simulation system, or we use the dead body of the human, the human dead body of the volunteers, or the dead animal which killed human in the human way. Or even we can send the student come to the clinical site for the observation. Alternatives in the research and testing is more interesting and diversified, and it will go through the different stage, like in vitro stage, in vivo, or in the silico or computer modeling. In vitro, the alternatives to animal testing can be found as the cell tissue culture techniques or physical chemical techniques. Regarding the cell and tissue culture technique, they usually, maybe I hope that you already heard about this before, because it's become very popular for maybe for a dozen years ago already. In this technique, the researcher will use the source of material mainly from the embryo or the stem cells. And one of the big advantage of this technique is that we can control many factors like temperature or the pH level, and it also gives us almost unlimited times of replication. It means that the source we can have the cells, the new cells or the new tissue unlimited and very fast, because that technique of the culture is give us the replication faster than we can have from the regular reproduction in the mice or rats or rabbit or any other creatures with a life cycle. The other one in vitro is the physical chemical techniques. One of them, one example for this one, for these alternatives technique is that chitosan films. These chitosan films can simulate the epidermal sheet, and it is used to in the testing of the formation of the drugs. It means that to let us know that how a substance or a drug are permeable through our skin. In vivo, the alternatives include the clinical skin pad test on human volunteer. This technique is you will see that there's the patch, the tiny patch, and each of them will contain a very small amount of the substance that we need to test, and we paste that patch under our skin, wait for a couple of time, or fast or long, half an hour or a couple of time. It will depend on each test, but the similar principle, and we will observe that how our body react to that drugs, or how we sensitive or not to that, or whether we allergy to that based on the expression on our skin. And because of it's a very small amount, so it's not really harmful to our body, but it still be able to tell you, to give us some clue about how our body to react to that. The other one, very interesting, is the 3D bioprint and the artificial organs. So far, the scientists already invented successfully a number of the artificial human organs, like lung, liver, cardiovascular system. And another one is I want to show you is that something called the biochips. The biochips can be DNA chips or organs on a chip or the microfluidic chip. Here's the picture of some of the artificial organs that I just mentioned, and here's the chip. You can see here's the an image of an organs on a chip. The organ on a chip was invented by the Wyss Institute for Biological and Bioengineering at Harvard University. It can replicate either lung or heart, intestine, kidney, bone marrow, or even blood-brain barrier. It's very, very complicated and very interesting, very useful. The DNA chips was mentioned, or also known as the DNA microarray. It was mentioned in one article of in the MIT Technology Review. The other alternatives is that for certain testing in silico or computer modeling is some program or model for analysis or programming or design based on computer. We can list out some here. For example, the quantitative structure activities, relationships, or computer-aided molecular drug design. And all of these, its principle is to base on the big data that we have nowadays and the greater capacity of the computer today. Without these two elements, all of computer-based alternatives will not exist. Although such achievement in technology to replace partially animal in testing, the alternatives we have so far still have some limitations. For example, it cannot mimic to give us the precise outcome in the behavior and psychology experiment, or it cannot tell us about the growth and aging progress. And for the computer-based program, because it's based on the past database from the information that we already collected. So if something unknown or any feature or asset that was not ever identified before, then such a new thing will become a missing piece in the puzzle. And it cannot tell us about the outcome in the genetic transformation or cell transplantation. A number of the scientists have seems very confident that we will soon to achieve the point of the time where the animal testing can completely eliminate it, such as the U.S. One example is that as David Green, an editor of the Science journal, already mentioned in one of his articles, that the U.S. Environmental Protection Agency already announced their plan that by 2025 they will reduce the request for testing and also the funding of such animal testing, especially on the mammal studies, by 30%, and will entirely eliminate such a study or request by 2035. And these ambitious timeline of the US EPA still receive the critics from a number of the scientists. So go go through this achievement is it gives us some idea about that how far we are, where are we now, and how uh, how how the progress of the development of the technique or the method, the approach method that we could obtain to so that one day we can eliminate, we can bypass totally the animal testing, and it seems that that future is not too far. It's a kind of the forcing future, and if that day come, we do not have to question, to wonder about that, which lives should be sacrificed, our human life or the animal life. That's that my presentation today. Thank you so much.